Welcome to our new video, that is MCQs on microbial growth and nutrition, and this is the part 1 of this video. So आइए देखते हैं कौन कौन सी MCQs है इन microbial growth and nutrition के ऊपर। तो फर्स्ट क्वेश्चन जो है वो है, growth in microbiology is referred to as increase in size, change in the population number, increase in cellular constituent, or all of the above. And the right answer about the growth in microbiology is all of the above. So माइक्रोबायोलॉजी में जो ग्रोथ का जो डेफिनेशन है वो ये सब डिफाइन करते हैं, जैसे कि इंक्रीज इन साइज, चेंज इन पॉपुलेशन नंबर, इंक्रीज इन सेलुलर कॉन्स्टिट्यूएंट्स, और denote the growth in microbiology or microorganisms. Okay, so move on to the next. It is known that growth of the bacteria is slower in the lower temperature. Choose the correct relation for the statement. And the first option is enzymes work less efficiently, lipids tend to harden and there is a loss of membrane fluidity, synthesis of protein is increased, both A and B. So the right answer about the growth of bacteria is slower in the lower temperature, that is option B, A and B. That is, enzymes work less efficiently when temperature is low, and lipid tends to harden and there is a loss of membrane fluidity when temperature is low. So these two is correct answer. Okay, so next question देखते हैं। एक मैथमेटिकल और न्यूमेरिकल क्वेश्चन है, यह है an inoculum of 10 to the power 7 bacterial cells was introduced into a flask of culture medium and growth monitored. No changes were seen in 18 minutes, that is the lag phase. Then growth occurred rapidly. So after a further 76 minutes, the population had increased to 4.32 into 10 to the power 8 cells. So what is the doubling time? So the options are 12 minutes, 15 minutes, 14 minutes and 20 minutes, and the right answer will be 14 minutes, and the mathematical expression is there. So 1 by t, so this td, that is the doubling time, and log n1 is the log n1 is the increased population, that is 4.32 into 10 to the power 8 cells. Okay, the ultimate population, and log n0 means the initial bacterial cells, so that is 10 to the power 7, and t minus d, that is the time taken to reach that doubling time, so that is the 76 minutes. So putting all these numbers, then you can solve this question. Okay, so ultimately the answer will be 14 minutes. Okay, so move on to the next question, that is, which of the following is not the characteristics of a growth curve? First is, shows development of microbial population under relatively stable environmental conditions. Next, plotted with logarithmic numbers. Next, graphs number of microbes versus time, number of microbes versus time, or each growth curve consists of 4 distinct phases. And the right answer about the growth curve of microorganisms is, each growth curve consists of 4 distinct phases. Okay, that is the lag phase, log phase, stationary phase and death phase. Okay, so move on to the next, that is, the intrinsic factor for the microbial growth is considered for which of the following factors? First one is nutrient content, next water activity, then protein content, and both A and B. And the right answer about the microbial growth depend on the factor, that is both A and B, that is nutrient content and water activity. Okay, so next देखते हैं कौन सी क्वेश्चन है, that is, in the growth of the microorganism, which phase is regarded as the period of the initial adjustment? That is lag phase, exponential phase, stationary phase, or none of the above. And the right answer about the growth of microorganism, that is lag phase. Okay, so the lag phase is the initial adjustment phase. So move on to the next, that is for the continuous growth of bacterial culture, which of the following method is mostly used? And first one is hemostat, microscope, culture culture, or chemostat. And the right answer about the continuous growth of bacterial culture is right answer is chemostat. So by this chemostat process, we can determine the bacterial. We can determine the bacterial culture in continuous growth of bacteria. Okay. So move on to the next. That is lithotrophs use which of the following compound as the electron donor for its growth and metabolism? And options are organic compound, NADPH, inorganic compound, and oxygen. And the right answer about the lithotrophs use as an electron donor, that is inorganic compounds. So lithotrophs use inorganic compounds as their electron donor. So next is the total biomass of an organism will be determined by the nutrient present in the lowest concentration relative to the organism's requirement is a statement of Lebesgue's law of minimum, or Shelford law of tolerance, or quorum sensing, or Heisenberg's principle of uncertainty. And the right answer about this statement is Lebe's law of minimum, and it stated that in environments where multiple nutrients are in low concentrations, the most limiting of these will determine the population density of the organism. So this is the Lebe's law of minimum. So move on to the next, that is choose the correct statement for turbidity measurement. Measure the decreased absorbance of the medium, measuring the degree to which a culture scatters light shown through it in a spectrophotometer. The growth is expressed as CFU per ml, or both B and C. And the right answer about the turbidity measurement is measuring the degree to which a culture scatters light shown through it in a spectrophotometer. Next is during exponential growth, the growth rate is number of generations per unit time, reciprocal of generation time, both A and B, and none of the above. And the right answer about the exponential growth, the growth rate is reciprocal of generation time. Okay, so move on to the next, that is the average size of the cells is lesser than the lag phase in which of the following phase of the microbial growth? And the options are exponential phase, stationary phase, death phase, or both A and B. And the right answer for the average size of the cells is lesser than the lag phase, in which it is exponential phase. So in exponential phase, the average size of the microorganism is lesser than the size when they are present in lag phase. So next one is choose the correct statement related to the stationary phase. So first one is secondary metabolite are produced, growth rate is same as generation time, also known as period of initial adjustment, none of the above. And the right answer about the stationary phase is secondary metabolites are produced in stationary phase. So this is the right answer. So next, choose the correct reason for the statement in stationary phase the reproduction rate is equal to the death rate. So the options are in stationary phase there is a complete cessation of division. Next, in stationary phase is limited by environmental factors. There is a fall of the viable cells, or both B and C. And the right answer is about the stationary phase, that is, in stationary phase there is a complete cessation of division. Okay, so move on to the next. Choose the correct statement for the stability of the thermophiles at the highest temperature. And the options are presence of inositol diphosphate and thermal stability of ribosomes. Thermal stability of ribosomes. Third option, presence of large number of polar amino acids and alpha helix protein. And the fourth option is increased leakage of cell components. And the right answer about the stability of thermophiles at the highest temperature is presence of inositol diphosphate and thermal stability of ribosomes. Okay, so move on to the next, that is bacterial cell growth depends upon a large number of cellular reaction of a wide variety of types. Choose the correct statement regarding this reaction. The reaction helps in transformation of the energy. Option B, such reaction result in the synthesis of the small molecules. Option C, it provides various cofactor for the growth of the bacterial cell. Option D, all of the above. And the right answer is option D, all of the above. Okay. So next is the feedset protein which is responsible for the division of bacterial cells. Which of the following organelles feedset protein is found? So option A is ribosome, option B mitochondria, option C chloroplast, option D both B and C. So where the feedset proteins are found. So feedset proteins are found in option B and C. That means in mitochondria and chloroplast. Okay. So next one is which of the following protein is responsible for the segregation of the replicated chromosome such that one copy is distributed to each daughter cells. So the options are option A MRIB protein, option B PAR protein, option C FITZ protein, option D both A and B. And the right answer about the segregation of the replicated chromosome, that is option A and B. Option A, that means MRIB and PAR proteins are responsible for the segregation of the replicated chromosome to each of the daughter cells. Okay, so next one is which of the following are functions of phosphate used in the preparation of culture media? So the option A is they act as a buffer and is a source of phosphorus. Next, act as reserve alkali. Next, source of phosphorus, or they act as buffer. And the right answer about the phosphate used in the preparation of media is they act as buffer and is a source of phosphorus. So next one is the isolation of the Neisseria meningitis by the use of certain antibiotics in media is an example of which of the following? The first one is selective media, option B differential media, option C enriched media, option D assay media, and the right answer is selective media. Okay, so move on to the next, that is which of the following cell function is seen during the stationary phase? First option A is cell cycle, option B energy metabolism, option C is biosynthetic process, option D both B and C. And the right answer about the cell function which is seen during the stationary phase, that is both B and C, that is energy metabolism and biosynthetic process. Okay, so move on to the next, that is Bacillus subtilis divides every 20 minutes. You inoculated a culture with exactly 100 bacterial cells. So after 4 hours, how many bacteria are present? And the options are 4,09,600 cells, or 4,096 cells, or 2048 cells, or 2 lakh 400 and 800 cells. And the right answer about this numerical question is option A, that is 4,09,600 cells. And how we can calculate it? So in 4 hours Bacillus subtilis will be divided 12 times, so therefore n is 12. So and 2 to the power 12 is equal to 4096. So total 100 bacteria present, so 100 into 4096 is equal to 409600 cells. So total 4,09,600 cell we will get. So move on to the next question, that is, it is also a numerical, that is, you performed a serial dilution and determined that the original number of cells in your sample was 12,000. How many bacteria will be present in 12 hours if the generation time is 15 minutes? Assume unlimited food and clean environment. Okay, so the first answer is 2.4 into 10 to the power 18, 3.4 into 10 to the power 18, 1.9 into 10 to the power 18, or 5.6 into 10 to the power 18. And the right answer is 3.4 into 10 to the power 18. So how we can get it? So number of generation in 12 hours is 4 generation per hour, that is, because 15 minutes is the generation time, so 4 hours, 4 generation per hour into 12 hour, so it's equal to 48 generation we will get in 12 hours. So therefore total number of bacteria in 12 hour equals to 12,000 into 2 to the power 48 equals to 3.4 into 10 to the power 18 bacterial cells. Okay, so move on to the next, that is, choose the correct function of the divisome protein ZIP-A present in E. coli is an anchor that connect the feeds it ring to the cytoplasmic membrane and stabilizes it. Next, helps to connect the feeds it ring to the cytoplasmic membrane. Next, recruiting other division proteins. And the last one, needed for peptidoglycan synthesis. And the right answer about the division protein ZIP-A present is an anchor that connects the fixed ring to the cytoplasmic membrane and stabilizes it. Okay, so the next one is, which is or are the dependent factor for the generation time of the given organism in culture? First option, that is option A, is the growth medium. Next, option B is incubation condition, then pH, and last one is both A and B. And the right answer about the factor for the generation time of the given organism, that is the both A and B. So the factor, that is growth medium and incubation condition, is required for the growth of the microorganisms. Okay, so these are the all MCQs in this part 1 video on microbial growth and nutrition. Okay, so thank you for watching this video.